metastasis, Laura will show that we put later on a second threshold here where this group has like hardly any risk of developing metastasis. I know she won't use that to me. But we set a threshold in this way. So the genes that are in those 70 genes are genes involved in the metastatic process. And you can see that the 70 genes are involved in all steps of leaving the primary tumor site, traveling through the bloodstream, and starting to build a new tumor elsewhere in the body. So I will not go into details of all our validation studies, but in the first one, consecutive series, we confirmed that if you have a good signature tumor, then the risk of dying of disease is very, very low. And developing a metastasis is also relatively low. And here we selected patients that in the past had only gotten surgery and did not receive chemotherapy. And as I just told you, that actually before 1995 that was more or less the standard for patients diagnosed with lymph node negative disease. So this is not like a rare outlier patient group, but in the Netherlands that was actually very common at that time. I got very, very many questions always, and first I sort of thought, where are these questions coming from? But only later realizing that the Netherlands is more or less more an outlier in being so conservative in chemotherapy as compared to other countries. So we also saw that already in small tumors, smaller than 50 millimeters, actually a proportion has already a high-risk signature. So that tells us that, like is seen by Chuck Peru and Teresa Surrey, that really the activity of genes is really intrinsic to how that tumor starts. So really from the start on 10 cells, 1 cell, it's already determined whether you're looking at a aggressive tumor or a much less aggressive tumor. So the diameter in its own is not sufficient. So we later validated that in 5 European hospitals under a European consortium called Transvic, where again patients who did not receive systemic treatment in the past in 5 countries showed similar outcome prediction. So in this case, if you compare that same patient population to what I showed you before, adjuvant online, where we also defined the clinical low-risk group as defined as predicted 10-year survival probability of more than 88% for estrogen receptor positive patients and only 2 for ER negative, we could compare the prediction of both using either one of those tools. First of all, in this same series, you can see that the proportion of high-risk patients by either one is a different proportion. So you would already reduce the number of low-risk patients by taking a gene profile. But also we observed that they assign patients as being high-risk and low-risk in a different way. So approximately 1/3 is discordant, which we have been observing in many other instances. And when we compared the prediction of outcome of disease by either the gene activity or the clinical pathological parameters, you can see that these curves are better separated here than here. So that's also, so first of all you assign less people to high risk by the 70 gene, but you also do it in a better way. Although there's still of course room for improvements. So if I showed you before that most guidelines would select overall in stage 1 and 2, 75% of patients with toxic chemotherapy, and that for 70% we have a risk to, the current clinical pathological risk assessment would actually over-assign many patients to high risk based on those factors by using tumor genomics, and this is also true for the other prognostic tests that I showed you. We could already reduce the number of patients and thereby reducing over and under treatment. So how, I think that one aspects of the talks of today is how does it implement in clinical practice. And I think one of the first studies we did is that we showed that in 16 community hospitals this test was being used, and this was a study sponsored by the Dutch Health Insurance Council, which is a government body in between the government and the private health insurance companies. So this council dictates to the private health insurance what they should mandatory insure in a basic package of insurance. And so they were interested to see how such a new molecular test would be used in clinical practice. So first of all, we compared risk assessment by the Dutch guidelines and the 70 gene signature and showed that 30% is discordant. And then in this particular study, where this was done on 427 patients, it was up to the physician and the patient to decide how to use the information. But it was assessed that 2/3 of those patients actually adapted their treatment advice based on the additional prognostic gene signature. So at the same time, we decided we wanted to make this test available to a more broader patient population, which actually then brings into place that you need to do technology assessment, but also get all sorts of quality controlled labels on such a test, which in Europe is actually only CE marking, which is more like technical registration. And we opted to have an ISO, which is like a CLIA laboratory certification. And for the US, this test got FDA approved and is now taken up in guidelines in the Netherlands and in European international guidelines. So this took a few, 2-3 years to set this all up, to make sure that when you do this once, that you will actually know what you're doing, and that such a test really gives reliable results that are given out in the same way over and over again. I think I may stop here and to show a few slides how the whole process takes with taking this all through the FDA, but I think I'll save this to a later time point. Take some questions.
Okay, some questions, Laura.
I guess, this is extremely useful for planning therapies. Do you see that in the future you might be able to use this kind of gene expression profiling while a patient is undergoing treatment in order to, modulate the drugs they're taking, or the timing or the dosage, and kind of adapt and see how they're responding?
I think this was one of the main reasons why I came here. To work on a clinical trial, and I think Laura will talk about it, I-SPY, where, so the test I showed you, MammaPrint, the 70 gene test, allows you to have prognostic information. But on top of that, one would like to give the information whether a tumor is responsive to a certain drug the patient will get. So those kinds of studies are done in, for instance, neoadjuvant trials like I-SPY. We give an example here. And I think that also allows you to identify whether a tumor with a certain gene makeup will respond to a certain drug. And if you would do that if you would have all that information, I think you may want to study the response of that patient by following them by imaging. But very early on is our indication that indeed you could have, after one cycle of chemotherapy, an idea whether that patient is responsive. So that's all still in a clinical trial, and it needs to be confirmed, so it's not yet used, but I think that's certainly where
So you think it's applicable not just for the clinical trials, but also for eventual clinical practice?
So it works in the
At
trial.
least also needs a little so first the clinical trials need to be completed. So we're working on it, the 3 of us actually.